Okay, so now we are going to continue from where we stopped last time. Last time we discussed about the classification of carbohydrates starting from monosaccharides, disaccharides, oligosaccharides, and polysaccharides. And remember, we said that polysaccharides are those sugars that cannot be hydrolyzed further into simpler forms, also called simple sugar. So in the case of simple sugar, in the case of simple sugar, the simple sugar, which is also considered as a monosaccharide, they are the class of sugars that they cannot be further split, that they cannot be further hydrolyzed into simpler units. So those types of sugar are called monosaccharide, or in the other term, we call them simple sugars. And they have a general formula. The general formula of these carbohydrates, sorry, that is the general formula for this monosaccharide is CN open bracket H2O, then close bracket N. So we are now also going to look at the classification of this monosaccharide on different bases. So we classified monosaccharide based on the number of carbon atoms, So we classified carbohydrate based on that. Then we also classified this monosaccharide on the basis of their functional group. That is those that have ketone functional group or those that have aldehyde functional group. So these are the basis of the classification of monosaccharides. So you have to know all this. The classification of monosaccharide is based on the number of carbon atoms or based on their functional group. So now we are going to look at this classification of monosaccharides based on the number of carbon. So those that have 3 carbon, they are called triuses. An example of these triuses, we have uh, glyceraldehydes. That is for those that have aldose functional group. And then for those that have ketones functional group, we have dihydroxyacetone phosphate. So that means that glyceraldehydes and dihydroxyacetone phosphates, they all have 3 carbon sugars, although they have different functional groups. Those, when we said aldoses, it means that are sugars that have aldehyde functional group, while ketoses are those that have ketone functional group. So glyceraldehyde and dihydroxyacetone are those different classes of sugars with different functional groups having 3 carbon. Then another types of these monosaccharide based on the carbon number, we have tetroses, that is those that have 4 carbon. So example of those sugars with 4 carbon under aldoses, we have what you call erythrose, and then we also have erythrolus. We have erythrulose. This erythrolus is also another examples of the tetroses, but under ketosis. Then we have under pentosis, we have riboses, we have ribose sugar. So ribose sugar is pentosis, that is those sugar that have 5 carbon. Then we also have xylose, then we have ribulose and xylulose under ketosis. Then we have hexoses, that is those sugars that have 6 sugars. So we have glucose and galactose under aldoses, then we have fructose. Then we have heptoses, that is those that have 7 sugars. Examples, we have glucoheptose and we have pseudoheptulose. So the heptulose is under ketosis. So ladies and gentlemen, you should all know that the monosaccharide can be defined or can be classified based on the number of carbon. And this is them. We have trioses, tetroses, pentoses, hexoses, and heptoses based on the number of carbon sugars. And also on that base, on that carbon number, they may also have different functional groups. So that is why for each and every one of them, they have aldoses and also ketoses. And then the next thing that we are going to look at is the typical example of these sugars based on the carbon number. So aldoses. So under this aldoses, under this aldehyde. So this is the glyceraldehyde. Look at it. We have 1, 2, 3. So we have 1, 2, 3. So we have 3 carbons. So based on that, look at it. The functional group is aldehyde. And remember, I told you when we are actually talking about this functional group, I told you that we have ketone functional group and aldehyde functional group. And one unique thing with this aldehyde functional group, the carbonyl carbon, and this is the carbonyl carbon. This carbonyl carbon is always having free hydrogen attached to it. So that is why it is aldehyde functional group. So we have glyceraldehyde. And look at also ribose. In the case of ribose, if you look at this carbonyl carbon, you will also observe that there is a free hydrogen. That is to tell you it is also aldehyde functional group. Then also for glucose, look at it also, we have the carbonyl carbon also have free hydrogen. So that is to tell you that glucose is also another example of aldehyde functional group. It's a sugar with aldehyde functional group. And this glucose is the most abundant monosaccharide in nature, is the most abundant. It has the highest percentage among all the sugars we have. And then we also have galactose. So, ladies and gentlemen, if you ask to give example of sugars that have aldehyde functional group, we have 4 here. We have glyceraldehyde, we have ribose, we have glucose, and we have galactose. All of them, they have aldehyde functional group. Then in the case of ketosis, we have dihydroxyacetone, we have ribulose, and we have fructose. So all of them, if you now look at it, you will see that in the carbonyl carbon, there is no free hydrogen. And remember when we discussed about the functional group of ketone functional group, I told you that there are two different alkyl group attached to it. We have 2 different alkyl group attached to it. So look at it. This is, you assume this as one of your alkyl group, and then you also assume this from here as also another alkyl group. So this is your carbonyl carbon. So it means that in your carbonyl carbon, there is no free hydrogen. So that is in the case of ketones functional group. So example of this ketones functional group, we have 3 of them here. We have dihydroxyacetone, we have ribulose, and we have fructose. So these are the examples of sugars with ketones functional group. So now the next thing is we are now going to look at some of the importance or some of the sugars of biological importance. Those sugars that are important in a biological system. So examples, we have trioses. So example of this trioses, remember I told you trioses from the previous slides, glyceraldehyde and dihydroxyacetone. They are examples of trioses, that is sugars with 3 carbon units. So, sorry, sugars with 3 carbon. So importance of this glyceraldehyde and dihydroxyacetone phosphate. 1, they are important as an intermediate of glycolysis. When we said intermediate of glycolysis, it means that they are very, very important in glycolysis. They are needed for the glycolysis to continue in the biological system. It means that along the way, when you take glucose for glycolysis, so at some point, these glyceraldehyde 3-phosphate are produced. And it's from this triacyl glycerol, it's from this glyceraldehyde that the glycolysis will continue. So it's an intermediate. It's important in glycolysis. And then also, they are also important as a precursor of glycerol. So this glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, they can be used in producing glycerol. And this glycerol is important for the synthesis of lipids in the body. So if you want to synthesize lipid in the body, you need this raw material. And this raw material is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. And these 2 trioses are the sugars that produce glycerol. And the glycerol is used to be esterified with different fatty acids to form fat and oil, which is lipids. And then the next sugars that is of biological importance, we have tetroses. Tetroses are sugars that have 4 carbon. So example, we have D-erythrose. So D-erythrose is also intermediate. It's an intermediate of carbohydrate metabolism. So it's important in carbohydrate metabolism. Then pentoses, that is those sugars that have 5 carbon. And examples, we have D-ribose. So D-ribose is a structural element of nucleic acid, RNA, and coenzymes. Remember from our previous classes, we talked about the component of nucleic acid. And one of these components of nucleic acid, we have ribose sugar and deoxyribose sugar. So that is why D-ribose is used as a structural element of nucleic acids. And remember, D-ribose is used for the synthesis of RNA, remember, while for the deoxyribose is used for the synthesis of DNA. So also at the same time, this ribose sugar it can also be used for the production of intermediates. And then the next thing also, we have, so the next types of sugar that we are going to look at is D-glucose. So D-glucose, fructose, galactose, and mannose, they are all hexoses. That is those sugars with 6 carbon. And for the glucose, it's used as a main sugar of the body. It is the main sugar of the body. Of course, it is the main sugar of the body because it's the glucose that you use as a starting material for glycolysis so that our body can provide or can produce enough energy. Then D-fructose. D-fructose also converted to glucose. It can be also converted to glucose and utilized by the body for energy generation. Then we also have D-galactose. It's synthesized in the mammary gland to make the lactose of the milk. Remember the lactose in the milk, the carbohydrates, the class of carbohydrates that are found in the milk of the breastfeeding mother is lactose. So lactose is the primary sugar or is the first important sugars that are consumed by the baby. So the D-galactose is important in the synthesis of the lactose that are found in the milk. And then we have D-mannose. So D-mannose is also a constituent of glycoproteins and glycolipids. And remember from our previous classes, I told you that glycoproteins and glycolipids are found on the cell membrane. They are found in the cell membrane. In fact, some of them, they serve as a receptor on the surface of the cell. So this is the sugars that are of biological importance. And then the next thing now that we are going to look at is the general properties of monosaccharides. Under the general properties of monosaccharides, we have the structure of glucose and fructose. We also have various forms of isomerism. General properties of monosaccharides. So number one, we have structure of glucose and fructose. We are going to look at them, and then we're also going to look at the various forms of isomerism. Under these various forms of isomerism, we are going to look at the D and L isomers. We are going to look at optical isomerism. We are also going to look at apimers, anomers, and then multi-rotation. These are what we are going to look at under this. So now we are starting with asymmetric carbon. Asymmetric carbon. What is asymmetric carbon? Whenever we say asymmetric carbon, it means it's a carbon that have 4 different groups attached to it. 4 different groups attached to it. Let's assume here is our carbon. From here, you can see that the color here, the color here, the color here, and the color here are all different. And that is to tell you that these groups, all these groups are different. They are not the same. So when we said asymmetric carbon, it means it's a carbon that have 4 different groups attached to it. Like, for example, let's say this is the carbon and we have hydrogen attached to it. And let's say here we have CH3. And let's say here we have NH3. And let's say here we have COOH. So if you look at this carbon, what is here and here and here are different. So all carbon, any carbon that have 4 different atoms or 4 different groups attached to it, that carbon is called asymmetric carbon. In the other term, you can also call it chiral carbon, or you can also call it asymmetric carbon or stereogenic carbon. You can call it asymmetric carbon or stereogenic carbon. So all those carbon that have 4 different groups attached to it is called asymmetric carbon. And another thing, no planar of symmetry. When we said no planar of symmetry, it means that they are not the same. They are not the same. And they have a mirror image. They have mirror image. Then it's non-superimposable. You cannot actually, if you cannot superimpose them. You cannot superimpose them. So they are mirror image. Like for example, now if you now look at your hand, if you now look at your hand, your left hand is the mirror image of your right hand. So that is how this asymmetric carbon look like. They have mirror image and they cannot be superimposed. So these are the general properties of those carbon that have 4 different groups attached to it. One, they are called asymmetric carbon. And why are they called asymmetric carbon? Is because they have 4 different groups attached to it. And as a result of that, they form mirror image. Like for example, look at it. This is the mirror image of this compound. This is the mirror. And this is the real compound, and this is its image here. So you cannot superimpose them. They are non-superimposable. And there is no planar of symmetry. So you should all know this. And another name for this asymmetric carbon, you can also call it chiral. You can call it chiral carbon, or you can even call it stereogenic. You can call it stereogenic. You can call it stereogenic carbon. So this is also another name for this asymmetric carbon. Okay, so you can actually have, and there is also a way that you can get the number of these isomers. You can get the number of the isomers a particular compound or a particular asymmetric compound may produce. We have this formula. We call it that is number of stereoisomers. So if you want to get the number of stereoisomers, you use this formula 2 power n. And the n means the number of stereogenic carbon. Like for example, in this compound, the number of stereogenic carbon, which is the number of asymmetric carbon, is 1. So it means that the number of stereoisomers is just 2 power 1, since the number of carbon is 1. So you can now say 2 power n. So 2 power n, the n here is 1. So you can now say 2 power 1. So 2 power 1 is just 2. So it means that they can only form two different structure. Remember from the word isomerism, when we said isomerism are compounds that have the same molecular formula but different structure. So if it is 2 power n, then n is 1, then it means that you have 2. So it means that compound, like this compound, can form only 2 different stereoisomers. Okay. And then now, so we are now going to look at pyranose and furanose ring structure. The reaction between the aldehyde group of carbon 1 with the alcohol group of carbon 4 or carbon 5 result in the formation of hemiacetal. So it means that in glucose, remember from where we discussed about glucose, in glucose, you see that we have aldehyde functional group. So the aldehyde functional group of carbon 1 is usually reacts, or it usually form a ring with either the OH group, that is alcoholic group of either carbon 4 or carbon 5. And this result in the formation of hemiacetal. And similarly, the reaction between the keto group of carbon 2 of fructose with the alcoholic group of carbon 5 result in the formation of a hemiketal. So ladies and gentlemen, you should understand this. Hemiacetal and hemiketal. In the case of hemiacetal, is the aldehydes, is the sugar with aldehyde functional group that react with alcoholic group of either carbon 4 or carbon 5 to form the hemiacetal. But in the case of hemiketal, is the keto group, that is ketone functional group of carbon 2 or fructose that react with the alcoholic group of carbon 5, resulting in the formation of a hemiketal. So the glucose exists in biological system not as a rectangle, but as a pyranose ring. So most of the glucose that are found in the biological system is in a form of the pyranose ring. So you should all know this. So how do we get this pyranose? So look at it, the formation of cyclic structure of glucose, that is aldoketose. Look at it here. This is where we have our aldehyde functional group from the glucose. And this is where we have OH group from carbon 5 or OH group from carbon 4. So if this form a ring, look at it, this carbon, this is the carbon 1, and this is the OH group. This is the OH group. So when this carbon 1 form or react with OH group of either carbon 4 or carbon 5, it forms alpha D glucopyranose. It forms alpha D glucopyranose or beta D glucopyranose. So now the question is, how did we differentiate this? What is the difference between beta D-glucopyranose and alpha D-glucopyranose? The difference is just this OH group, orientation of OH group. So in the case of alpha D-glucopyranose, the OH group of the anomeric carbon is looking, or is actually looking downward. It is looking downward, while for the beta, it is upward. It is upward. So this is how to differentiate the beta D-glucopyranose and alpha D-glucopyranose. And the ring structure of this glucose is called pyran. It's called pyran. Okay, then I think this one is clear. Then, so the next thing now that we are going to look at is in the case of the hemiketal, that is when we are going to have a cyclic form of fructose. So you see, this is your carbon 2 of the ketone functional group, then it now reacts with carbon 5. Then as a result of that, we also have alpha D fructopyranose. And look at, in the case of alpha, what is the difference? It's still the OH group. So the OH group here is downward in the case of alpha, while the OH group is upward. So that is why they are differ. One is alpha and one is beta. So just always to differentiate between alpha and beta, you always try to differentiate the orientation of the OH group that are found in the ring structure. And then the next thing is we are going to look at the reference carbon form of sugars. This is the glucose, and this is the glucose. But what is the difference between the two? One is D-glucose and one is alpha glucose. So what is the difference between alpha and D-glucose? So at carbon 5, in the case of D-glucose, the OH group is by the right, while in the case of L-glucose, the OH group is by the left. So that is how this sugar differ. So you see, now we talk about two things, alpha and beta glucose. How alpha and beta glucose differ with one